Hello dear students, today we'll explain a new activity in Chapter 1. Activity 2, Chemical Transformation of Food, page 18-19. As we know in Activity 1, as a definition of digestion, digestion is the transformation of large food molecules into simpler ones and finally into nutrients. Here we should distinguish between digestion in vitro and digestion in vivo. What do you mean by digestion in vitro? Means digestion inside lab, outside the body, but keeping same conditions as within body. What do we mean by digestion in vivo? Means digestion inside the body of the animal. What do we mean by experimental setup? Is the group of tools or glassware used during an experiment in the lab. Here there is a video about the enzyme. Each enzyme has an active site, and this active site is the binding site of the substrate. When the substrate binds to the enzyme or the active site of the enzyme, the substrate is digested into simpler ones or into simpler molecules. As a conclusion, substrate is a food substance upon which the enzyme acts. Example, starch, proteins. Whereas the enzyme is like the scissor in the adjacent figure, which cuts or digests the substrate into smaller molecules. Means enzyme is a chemical substance or biological catalyst that breaks down large food molecule of substrate into smaller ones and finally into nutrients. In the activity in your book, document A and B show an experiment about the digestion of cooked starch. Both tubes A and B are placed in a water bath at 37 degrees Celsius. Both tubes A and B contain cooked starch, but Tube A contains, in addition to cooked starch, a small amount of fresh saliva. Then, two tests are performed, iodine test and Fehling test. For iodine test, at the beginning of the experiment at t equals 0 minute, both tubes give dark blue color, whereas at the end of the experiment at t equals 15 minute, in tube A there is orange color, whereas in tube B there is dark blue color. For Fehling test, both tubes A and B give dark blue color at the beginning of the experiment, whereas at the end of the experiment at t equals 15 minutes, in tube A it gives brick red precipitate, whereas in tube B it gives dark blue color. This means that the starch in tube A transforms into sugar or digests into sugar. As a conclusion, by the action of the saliva, the starch in tube A is digested into sugar. Finally, as a conclusion, salivary amylase, which is an enzyme, digests starch into simpler sugar, maltose, at 37 degrees Celsius in the mouth. In the mouth there is a medium which is characterized by the pH equals 7, so it is a neutral medium and not acidic medium. Acidic medium only found in the stomach. The conditions of salivary amylase or saliva are substrate, starch, since it acts on a specific substrate which is starch and it digests it into sugar. Medium, neutral, pH equals 7 in the mouth. Duration, 15 minutes. It takes 15 minutes to digest starch into maltose. And at the body temperature 37 degrees Celsius. This digestion is called hydrolysis. What do we mean by hydrolysis? Hydro is related to water. Lysis, it means digestion. So, it is the digestion of complex food molecules into simpler ones in the presence of water. This means that salivary amylase digests starch into simpler sugar maltose in the presence of water.